Hello everyone. Before this video gets started, I just wanted to remind everyone that our free online tutoring for the upcoming semester is starting soon. So head over to studystem.org if you need help with chemistry, physics, or biology. Thank you and enjoy the video.
So just a quick summary of this big question. The question just mentions polycystic kidney disease. It gives a bit of an introduction to what polycystic kidney disease is, and then relates the sodium-potassium pump with the steroid hormone albumin. And in the end, it produces a hypothesis that a signal transduction pathway that includes the protein kinases MEK and ERK may play a role in causing polycystic kidney disease. Now I'm going to be explaining this diagram here. So you can see that the sodium-potassium pump is embedded in the plasma membrane, and bound to the sodium-potassium pump is the steroid hormone albumin. Now after that, the signal transduction pathway is being carried out, which is involving two kinase proteins, namely MEK and ERK. MEK stands for mitogen activated protein kinase and ERK stands for extracellular protein kinase. Both of them have their roles in vasoconstriction and growth of vascular smooth muscles. And apparently scientists think, according to this question, that the polycystic kidney disease is caused by the presence of albumin and its binding with the sodium-potassium pump. So part 1 says, describe the characteristics of the plasma membrane that prevent simple diffusion of sodium and potassium ions across the membrane. Explain why ATP is required for the activity of the sodium-potassium ATPase. Now, the plasma membrane is a semi-permeable membrane, which means that it only allows selective particles to pass through it. And the plasma membrane, its interior is hydrophobic or non-polar, whereas its exterior is hydrophilic or polar, in other words. These properties prevent charged particles from entering or passing through the plasma membrane by simple methods like diffusion. So the sodium and potassium ions will have to find some other way to cross the plasma membrane. In this case, that other method is the sodium-potassium pump. Sodium-potassium pump. The sodium-potassium pump pushes the sodium and potassium ions against their concentration gradients by using ATPs as a source of energy. Now, suppose that this is the plasma membrane wall, and suppose that this is a sodium-potassium pump embedded inside the plasma membrane wall. Now, how this works is that the sodium-potassium pump, this pump will use ATPs as a source of energy to push the sodium ions from an area where they are lower in concentration to an area where they are higher in concentration. Most commonly, sodium ions using the potassium-sodium pump would be moving out of the cell, whereas the potassium ions, they would be moving inside the cell where they are found in a higher concentration. Next part, identify a dependent variable in the experiment represented in figure 1. Justify the use of normal human kidney cells as a control in the experiments and justify the use of a range of albumin concentrations in the experiment represented in figure 1. So figure 1 would be this figure on the left hand side. Now, by observing this figure, you can clearly tell that the dependent variable in this experiment was the number of cells. The dependent variable is the one that depends on a number of different factors in that experiment. For example, if we were to use a large number of cells in this experiment, And if we were to use a small amount of albumin for that large amount of cells, then we wouldn't be able to properly identify the number of cells that developed the disease PKD. So the dependent variable here would be the number of cells. The next part says justify the use of normal human kidney cells as a control in the experiments. So the normal human kidney cells have been used as a control in these experiments so that the scientists can compare the normal human kidney cells with the PKD cells, with the cells that develop the disease. And by doing so, they will be able to determine the effects that are produced by this PKD on the normal human kidney cells. As for the last part of this question, justify the use of a range of albumin concentrations in the experiment represented in figure 1. So as we saw the figure 1 before, this is figure 1. It shows the relationship between the number of cells and albumin concentrations in PM. Now, the purpose for using a range of albumin concentrations was to identify which concentrations of albumin were affecting the cells, which concentrations of albumin were developing that PKD disease in the normal human kidney cells. So the next part says, based on the data shown in figure 2, describe the relationship between the concentration of albumin and the sodium-potassium ATPase activity, both in normal human kidney cells and in PKD cells. The scientists determined that the sodium-potassium ATPase activity in PKD cells treated with 1 pm albumin is 150 units of ATP hydrolyzed per second. Calculate the expected sodium-potassium ATPase activity in units per second in PKD cells treated with 10 to the power 6 pm of albumin. So, the relationship, the first part of the question, tells us to describe the relationship between the concentration of albumin and the sodium-potassium ATPase. So clearly, as the concentration of the albumin is increasing, the activity of the ATPase, of the sodium-potassium ionic ATPase, is decreasing. So that means that albumin concentration has an inverse relation with the ATPase activity, or a negative correlation, in other words. Or in other words, you could say with the increase in albumin concentration, the activity of the ATPase is decreasing. As for the second part of this question, to calculate the expected sodium-potassium ATPase activity in units per second in PKD cells, we're going to have to use this graph as well as the data given in the question up above. Now the data given in the question says that the albumin concentration they have given us is 1 pm, and that 1 pm albumin concentration has hydrolyzed 150 units per second of ATPs. What they're asking us is what amount of ATPs will be hydrolyzed per second when an albumin concentration of 10 to the power 6 pm is used. So we're going to have to compare this data we have with the graph that we have. The graphical data suggests that at 1 pm concentration of albumin, the ATPase activity is approximately 100%. And at 10 to the power 6 pm albumin concentration, the ATPase activity drops down by a 70% and reaches approximately 30%. So this means that if the 1 pm concentration of albumin leads to the hydrolyzation of 150 units per second of ATP, then 10 to the power 6 pm albumin concentration should decrease the hydrolyzation of ATPs by 70%. So this means what we get left at a 10 to the power 6 pm albumin concentration would be 30% of the original ATP amount hydrolyzed. So 30% of 150, that would be approximately 45 units of ATP hydrolyzed per second. The fourth part of this question starts with, in a third experiment, the scientists added an inhibitor of phosphorylated MEK to the PKD cells exposed to 10 to the power 4 pm albumin. Based on figure 3, which is this figure, predict the change in the relative ratio of ERK to PERK in albumin-treated PKD cells with the inhibitor compared with albumin-treated PKD cells without the inhibitor. So now we have a comparison between two PKD cells, one of which has an inhibitor in it and the other one which does not have an inhibitor. An inhibitor, as you might already know, is something that decreases the activity of enzymes. An inhibitor decreases activity of enzymes. And the enzymes that it's going to decrease the activity of, in this case, will be MEK and ERK, both of which are a part of this whole process. MEK stands for mitogen protein kinase, whereas ERK stands for extracellular signal regulating kinase. Now, the whole process is dependent on this inhibitor. The relationship, the comparison the whole thing will be dependent on this inhibitor. Because if the inhibitor is present in this reaction, then the MEK enzyme will be unable to function, and that means that no more phosphorylated MEK will be made. If the phosphorylated MEK is not being formed, then obviously the phosphorylated ERK will not be formed either, because as you can see in the diagram, the reaction involving the phosphorylated MEK and the reaction involving the phosphorylated ERK are related with one another. And you need the phosphorylated MEK to make the phosphorylated ERK. So if the phosphorylated MEK is not being formed, the phosphorylated ERK will not be formed either. So this will be the situation for the PKD cell that has an inhibitor. As for the PKD cell that does not have an inhibitor, both the phosphorylated MEK as well as the phosphorylated ERK will be formed at the same time. Now, back to the question. It says to predict the change in the relative ratio of ERK to PERK. So the possible answer here would be that the ratio of ERK to PERK will increase in the cells with the inhibitor, which means that the ERK compared to the PERK, the ERK will increase, whereas the PERK will decrease, as no more PERK will be formed in the cell that has an inhibitor in it. But the cell will continue producing ERK, as there is nothing stopping the production of ERK itself. Next, it's asking us to justify our prediction. The prediction that we made was that ERK ratio PERK will increase. Why? Because the amount of PERK is not increasing, instead it is decreasing, but the amount of ERK is increasing. And this is the situation for the cell, for the PKD cell that has an inhibitor in it. And the reasoning behind this, we also discussed that. The reasoning is that since there is an inhibitor attached to the MEK enzyme, then that means no more phosphorylated MEK is going to be formed. And if no more phosphorylated MEK is being formed, then that means no more phosphorylated ERK will be formed either, because as you can see in the reaction pathway here, the formation of phosphorylated ERK requires the presence of phosphorylated MEK. So without the phosphorylated MEK, phosphorylated ERK will not be formed. Yet the formation of ERK will still continue because there is nothing stopping the production of ERK itself. The next part says to explain how the concentration of cyclin proteins may increase in PKD cells treated with 10 to the power 4 pm albedo. As you can see in the figure 1 of this question, when the concentration of albumin is 10 to the power 4, the number of cells is maximum. So, the signaling pathway, this pathway will stimulate the transcription of genes involved in cellular division. And the target genes, they will likely include those for cyclin proteins because cyclin proteins are the ones that regulate the cell cycle.